I was really happy to hear that I was able to give this talk today for the simple reason that I get to discuss, the fine details of project design, which is something that I definitely enjoy as an experimentalist. What I'm going to talk about today is discussing mutational scanning library construction. It's certainly an important and necessary part of many of our projects, many of which we heard about in this meeting. But the details about choosing a library prep method and executing it are often only briefly mentioned in the main body of a publication. So with that said, today I'm going to go over some examples of mutagenic library prep approaches and also compare each of these. Also acknowledging that what I'm presenting is certainly not an exhaustive list, and I certainly hope not to offend too many of you whose favorite methods I'm not going to be mentioning. I'm going to go into deeper detail on the method that we use in the Roth lab called Popcode and how we've recently made it even faster. And lastly, I do want to go over some other factors that are certainly important when designing your library prep strategy. Before that, I think the first tip for overall success in designing your library prep approach is to have a clear idea of the goals that you want to achieve. So some aspects to consider are properly defining the length of the region you want to mutate, whether your assay will benefit or be hindered by having one or more than one mutation per clone. Will it benefit from measuring effects from multinucleotide variants, or does your assay tolerate or require lots of wild type or completely non-functional variants in the screen? And these, could either arise from nonsense mutations or from, frame shifts or deletions. And lastly, are you only interested in a very defined set of substitutions, or do you want to focus, on having, as many different types of mutations as possible? Right. So I'll present some methods now and, noting as best as I can how they might fit in with each of these considerations. So the first method I'll discuss is error-prone PCR, which is likely to be the oldest and probably still the most widely used method. It's the most popular version of this is to simply use a mutant of Taq polymerase, which has a higher misincorporation rate. And you can tune the mutation frequency by playing around with other factors, like the addition of manganese, using higher PCR cycles, or using an unbalanced pool of DNTPs. So you can also mutagenize DNA by incorporating oxidized nucleotides into your PCR. It's certainly straightforward approach. All you have to do is incubate DNTPs with an oxidizing agent like ferrous oxide, and then add these into a PCR reaction directly. And essentially, these oxidized nucleotides are incorporated into the DNA, and then replication by Taq polymerase at an oxidized nucleotide leads to a misincorporation event. So the main disadvantage to these protocols is that they are extremely biased for single nucleotide variants, and of these, they're highly enriched for transition events. So in the Roth lab, we, one of our first libraries that we generated was using the oxidized nucleotide approach. So we generated and sequenced about 4,000 variant clones for human UBE2I, which is a sumo-conjugating enzyme. So on the right here, I'm showing the distribution of clones carrying various numbers of missense mutations, as well as the proportion of clones that were either wild type, carried a frame shift, or nonsense mutation. And on the bottom, just showing for each amino acid position of UBE2I, the fraction of observed missense mutations that are achievable with a single nucleotide change, or those that require 2 to 3 nucleotide changes. So you can clearly see that there are nearly no SNP inaccessible changes observed in any of these clones. Overall, in the 4,000 clones that we sequenced, we only observed 27% of all possible amino acid changes. So in summary, for error-prone PCR, it's certainly a very simple approach to employ. It requires very little in terms of reagents, but I certainly would stay away from it, if you're interested in exploring the full spectrum of coding variation in your screen. The remaining methods that I'm going to go over all rely on the use of oligos to introduce variation. And the first option that you can choose for ordering your oligos is called doped oligos or spiked oligos. Essentially, you order an oligo and indicate that you want a certain amount of error at each of the nucleotides. The oligo type that we tend to see more in full saturation libraries is the use of, I just call them codon series oligos. So essentially what you do is design an oligo for every codon that you want to mutate and replace the codon with either a completely degenerate NNN or something that's more refined like NNK or NNS, which they'll still generate all possible amino acids, but you'll only generate one of the 3 stop codons. Lastly, if you want to eliminate, certain types of variation entirely or generate a very precise set of variants, you can order custom oligo arrays. So when using oligos, you may still observe bias towards single nucleotide variants, especially when your library prep protocol involves hybridizing oligos to a template, albeit it's certainly much less biased than error-prone PCR. We do observe a certain amino acid that at certain amino acid positions, there can be more or less frequency of changes. And we found that a major factor that influences this is actually, just the GC content surrounding that codon. So I highly recommend using a primer design tool to help alleviate this bias. So for example, we have one that was designed by Jochen Weile in the lab, and it specifically designs oligos with central degeneracies. So if you want to mutate, a relatively small region, a very straightforward method like fragment assembly could be used to create a library. So all you have to do is, create a pool of double-strand oligos or short DNA fragments that contain, variants that you want, and then introduce these to a plasmid using something like Gibson assembly or perhaps ligation after cutting with a type 2 restriction site. This method, it is limited to the size of the oligo that you can synthesize, and with the caveat that the longer the oligo, the more prone your library is to be contaminated with truncations or deletions. It certainly is nice because it is a one-tube method, and you can easily tune the number of mutations you want in a single clone just by ordering whatever pool of oligos that you want. In overlap extension PCR, a combination of overlapping doped oligos can be combined with a molecular excess of outermost oligos, and they essentially just use each other to prime synthesis, and after PCR you end up with a longer fragment that can then later be cloned into an expression vector, for instance. Similar to more simple assembly methods that I just mentioned, these can contain many deletions again, and they may best be suited for screening where you want to identify mutations or variants that convey a certain phenotype that you can easily pick up in a pooled assay, for instance. It's also amenable to be done in a single tube, which is nice, and it is relatively straightforward in terms of, hands-on time. You're essentially just doing a PCR. The oligo design may be the limiting factor here that will affect the final results, so you may need to play around with, number and types of oligos that you're using. The next method that I wanted to touch on is, I think is used a lot, which is inverse PCR. This method is great if you're really adamant about having just one individual mutation per clone that you generate. So the process is very straightforward. You simply prime a PCR reaction with an oligo that contains a 5' mutation, such as like a degenerate codon, for instance. And the second reverse primer is immediately flanking the other one just in the opposite direction. So after PCR, you essentially create a linear plasmid, then that you just, phosphorylate, ligate, and transform. Now, because each codon requires its own unique set of oligos, it can't be done in a single tube, at least not that I'm aware of. Which I think is the main caveat of this method. But, the overall method is very straightforward and could be done essentially by anyone that can do, a plate of PCR reactions. So I'm going to touch on some more popular one-tube saturated mutagenesis methods, the first one being Fungal. So it requires the synthesis of a urisylated single-strand template in bacteria, and then annealing mutagenic oligo to it, and then this oligo is then extended, and then the nick is then sealed by ligation. The strands are then denatured, and then a second universal primer is added, and which is used to prime synthesis of the second complementary strand. After this, the urisylated template can then be degraded using a combination of uracil DNA glycosylase and exonuclease. A second very similar method uses DNA nicking at a specific restriction site. So you selectively nick the top strand of your duplex and then degrade it using exonucleases. Mutagenic oligos are then annealed, extended, and then, the nicks are then sealed by DNA ligase. Bottom strand is then nicked and again removed by exonuclease digestion. A common oligo is then annealed and extended, and then this, creates your new double-strand mutated plasmid. I think these methods are very powerful one-pot methods for doing saturated mutagenesis. And apparently, I've never tried them myself, but apparently you can modify the protocols in order to tune the number of mutations that you want per clone. Now, the major disadvantage to them is that they do require your plasmids to have unique sequence features on them to perform the assays. They do also require many molecular biology steps, so it's quite possible that more troubleshooting may be required to execute them successfully, especially if you're attempting to use them for the first time. Another relatively new method that has been developed is called Programmic allelic series. It also requires a single-strand urisylated template. So in the first step, it just uses a urisylated forward strand. And to this is annealed a 5' tagged mutagenic oligo. This oligo is extended by DNA polymerase, and then the urisylated template is removed using UDG and exonuclease. So the tag that was introduced on the oligo is then used to prime PCR amplification of the newly created strands. The tag is then cleaved using type 2 restriction site. Now, the newly created products are then used as megaprimers, which then are bound to a new bottom strand that is that is also urysylated. So this mega primer is extended to the end of the template, and again, the bottom urysylated strand is again removed using UDG and exonuclease, and then the final full-length products can then be PCR amplified. So again, this protocol does generate a large number of clones that have single variants, and what's nice about it is that it doesn't require any special, DNA sequences or features on your starting material, but you do have to order oligos that have, these unique 5' tag sequences on them. Similarly to the other one-pot methods, it does have many molecular biology steps in it, and again, may take some time to optimize, if this is the first time that you're trying to use something like this. So the last method I want to present is the one that I and others developed in the Roth lab called Popcode, where essentially we borrowed from various other mutagenesis strategies to, design a protocol that, works well for us in the lab. So in its, original version, we also used a urysylated template that was generated by PCR, just with Taq DNA polymerase that could incorporate DUTPs. So it's denatured, and we combine it with a pool of mutagenic oligos and also a molar excess of a 5' oligo. And this is incubated with a non-strand displacing polymerase and Taq DNA ligase to extend the gaps and seal in the nicks. Then the urysylated template is degraded by UDG and heat treatment. And then, the remaining newly generated strands are then amplified by PCR. In its kind of newer second version, we removed the urysylated template, and now we just use our, denatured wild-type plasmid as a template. And again, mutagenic oligos and a 5' oligo now with a tag sequence are annealed and extended. And after that, we specifically amplify the newly created mutant strands simply by priming PCR on the tag sequence that was introduced on the 5' end of those molecules. And then, these can be directly cloned and sequenced after that. So in terms of ease of use, tag Popcode requires relatively few molecular biology steps. And, it also doesn't require any special features on your DNA or your oligos. I certainly wouldn't recommend it if, you're interested in generating clones that only have single missense mutants in them, though. Popcode is definitely a great choice if you want to generate libraries containing all possible coding variants. So just as an example here, I'm showing a tag Popcode library that I generated for the LDL receptor. So I'm only showing you data for the first 150 amino acids, just for simplicity. So the x-axis here shows the amino acid position, and the y-axis shows the amino acid residue that's observed in this library. And orange is just an indicator of the frequency that amino acid change is observed in this pool. So, I think this, this figure shows that, we are observing nearly all amino acid changes in a relatively equal amount in this library, which is nice. So for longer open reading frames, we generally separate our mutation space into regions when we're performing Popcode. So here for LDLR, I generated 5 separate libraries where each region, and a typical region will span 150 to 185 amino acids, is mutated, and the rest, we don't touch. It just remains wild type. The reason that we do this is, simply because it allows us to more tightly control the overall density and, number of mutations in a single clone. So I'm just showing an example on the right here for LDLR region 1. And this shows the distribution of clones that contain, various numbers of missense mutations in green. And in this particular library, there's a mean of approximately 1.2 mutations per clone. So lastly, I just want to touch on a couple other factors that I think are important to consider when designing your library prep strategy. So the first being, whether or not you should add DNA barcodes to your libraries. I would recommend doing this if, the length or the length of the DNA that you want to query is relatively long. And this because it allows you to perform, really straightforward short read sequencing of the barcode to, identify and count variants, after when you're performing your screens. So you don't have to go through, you do have to go through the effort of, characterizing your library before you're performing screens, which is going to require some type of long read sequencing like, smart sequencing or nanopore sequencing, because you do need to associate the sequence of each of the barcodes with, the variant genotype. The length of your barcode and the composition of your barcode, that can be dictated by the number of clones in your library. So I typically use 25 base pair barcodes since, our libraries contain, roughly 200,000 clones. I also alternate strong and weak bases at my barcodes. And what that allows is for each barcode to have, roughly a similar GC content and similar melting temperature. And the reason for that is to help reduce PCR amplification bias of individual barcodes. So lastly, I just want to touch on complexity. And, what I mean here is, maintaining the overall abundance of clones or the overall number of variants throughout all the processes of, your library prep and screen. So you want to essentially minimize the opportunity where, you could introduce, a bottleneck where a significant portion of your clones or variants are going to, drop out of your library. This can arise at multiple stages. One example here is just at the step where you're introducing your library into your plasmid. So you want to ensure that the strategy for generating the plasmid pool actually, generates enough single events to capture all of your variants. So you can do this in a number of ways. So we use gateway recombination sites, to efficiently recombine our linear fragments into a plasmid. But you could use, Gibson assembly or even restriction enzyme cloning, just making sure in all cases that you're scaling it up appropriately. You also want to ensure that you are transforming enough of this product into bacteria, and to track the number of individual transformation clones you generate, and make sure that it's large enough to encompass all of the variation that you introduced at your library prep stage. Although there may be instances where you want to purposefully place a bottleneck or cap on the number of clones in your library, you may want a defined number of clones in order to be able to confidently detect all of them in your screen. So you could do this simply by, tuning your bacterial transformation efficiency and pooling a very, defined number of colonies that you count. So lastly, I would finally suggest that, if you're new to library preparation or are trying out a new method, that you maybe try a couple methods at the same time, to increase the chances that one of them will end up working for you. I think it makes sense to kind of, start with a broader horizon and then, hone in and run with a method that, gives you the results that you want the most easily. You're also not restricted to using a method exactly as it's published. Our Popcode method is certainly a mashup of various methods taken from other techniques, and you can surely do the same thing, to customize a method based on your needs. So with that said, I would like to thank others in the Roth Lab who were involved in developing our mutational library prep methods, specifically Nishka, Jennifer, Song, Jochen, and Marta. Thank you.